This video is going to be a quick overview of mitosis. So there are a few reasons that cells need to divide. So because we need new cells for growth or to replace old or damaged cells, we're going to go through the process of mitosis. We need to make sure that our new cells are identical copies of the original cells. So one of the first things that cells must do is they must make a copy of their DNA. Every cell needs to get a full set that is identical to the original DNA. So DNA actually opens up and unwinds, which we can see here in this image. This provides a template for new pieces of DNA to come in so that now we can have two strands of DNA so that then we have an identical copy of the original DNA that we began with. The majority of the cell cycle is interphase, which is about 75 to 90% of the time that cells spend in this time of interphase here. So there are actually three parts to interphase, G1, S, and G2. During the G1 phase, the cell is growing and preparing, making copies of its organelles and getting ready to prepare to be able to divide. During the S stage is when the cell makes a copy of the DNA. And then finally, now the nucleus can begin to break down and the cell further grows larger in order to prepare to divide. Mitosis is actually very short. Cells do not spend a lot of time in these phases. The phases of mitosis include prophase, metaphase, anaphase, and telophase. So we'll go through each of these parts of mitosis in order to end up with cytokinesis, which is going to be that final division of our cell. So as I said, there are three parts to interphase. First is the G1 stage, in which the cell grows and matures and makes more organelles and simply carries out its normal activities. After that, it's going to make a copy of its DNA so that way there is a full set of DNA for each of the new cells. Still in part of interphase, we finish with the last stage, G2, where the cell grows through more growth, the DNA is already copied, and we're just making sure that we've got all the organelles and structures needed to do mitosis. This is what a cell looks like in interphase. We can see the nucleus is still very well condensed, and we really can't see individual chromosomes. Most of the cell's life is spent in interphase. So when we look at cells under a microscope, most of them are going to be in interphase. The process of cell division is called mitosis. In mitosis, we start with prophase. During prophase, we begin to be able to see a little bit of the chromosomes, and the nucleus is broken down, and we start to see the nucleolus start to break down, and we can see that we can start to see those chromosomes. After prophase comes metaphase, in which the chromosomes are lined up in the middle, the mitotic spindles attach so that they're ready to pull the chromosomes apart. The centrioles are on opposite sides, and the chromosomes are all neatly arranged. Next, we go through anaphase, in which the sister chromatids are pulled to opposite sides of the cell. In anaphase, it's very important that there is an exact copy of DNA being pulled to one side and an exact copy of DNA being pulled to the other side. After anaphase is telophase, in which our sister chromatids are at opposite sides, the spindles disassemble, and we begin to see the development of the nuclear envelope around the sister chromatids again, and we might begin to see the beginning of cytokinesis. For comparison, you can see in anaphase the chromosomes are pulling apart, versus telophase the chromosomes are on opposite sides and they're really beginning to condense and form into two separate nuclei again. Finally, we have cytokinesis, which means the cytoplasm is going to divide. Our cell is going to go and divide into two identical halves called daughter cells. If it's a plant cell, we have a cell plate that forms. If it's an animal cell, then we have a cleavage furrow that forms to split the cell.